Bismillahirrahmanirrahim. My name is Hidayatullah, lecturer in botany, Government Degree College, Mirpur. Dear student, today we discuss about chapter number 6, Kingdom Prokaryote. Monera. Kingdom Prokaryote, basically, we contains only prokaryotes. Prokaryotes are those cells which lack true nucleus and membrane-bounded organelles. From prokaryotes, eukaryotes have evolved, and eukaryote is advanced. Eukaryotes contains protopista, fungi, plants, and animals. While we are discussing about prokaryotes, prokaryotes are primitive. Prokaryotes contains two main domains, archaebacteria and eubacteria. Archaebacteria is basically ancient bacteria. These bacteria can live within extreme environment. They have high tolerance level. Because of that tolerance, they are known as halophiles, thermophiles, and methanogens. Halophiles means they have high level of salt tolerance. Thermophiles means they have high level of temperature tolerance. Similarly, they produce methane as their metabolic products. So that's why they are known as methanogens. While we are discussing about eubacteria, they live everywhere. They may be gram positive, gram negative. So these bacteria are simple microorganisms, unicellular. They may live in association and they form colonies. If we differentiate archaebacteria and eubacteria, we can differ them by their DNA and RNA polymerase, and respond to their antibiotics. Dear student, now we discuss about discovery. Bacteria was discovered by Anton van Leeuwenhoek in 1676. When he discovered them, he called them animalcules. Animalcule means protozoa and bacteria. When he was observing through his simple macroscope, he first observed a drop of rainwater. He saw some tiny objects which were wriggling and dotting within that drop of water. Then he also observed vinegar, saliva, and other substances. Then he wrote letter to Royal Society of London about his discovery. So, after Anton van Leeuwenhoek, Pasteur, Robert Koch also worked on these macros. Now we discuss about occurrence. Occurrence means bacteria are found everywhere. That's why we are calling them cosmopolitan. Bacteria are present in air, land, water, oil, food, decaying organic matter, plants, man, animals, hot springs or vents, or they are also found even though in freezing temperature. While we are discussing about their size, they are very small macroorganism. If we discuss about their width, that is 0.2 micron to 2 micron, length 2 micron to 10 micron. Now we discuss about its structure. This is very simple structure. We are observing here there is nuclei, cytoplasm and ribosomes, pili, chromatin body, plasma membrane, cell wall, capsule, plasmid. These are the components we will discuss one by one. The first one, that is cytoplasm. Cytoplasm is basically inside the area inside the plasma membrane, that is granular form. This cytoplasm contains small vacuoles and granules of glycogens, proteins, fats, even though ribosomes are present which are in scattered form. And there are small food vacuoles are also present within cytoplasm. Now we discuss about chromatin bodies, that is also known as nuclei, because bacteria lack true nucleus. There is no nuclear envelope, there is no nucleosomes. That's why we call it nucleoid. So bacteria contain that chromatin bodies as a single DNA that is scattered within the cytoplasm. And this DNA controls all the regulation or regulatory activities. Then comes outside, look here, these are the pili. Pili basically small halo appendages, and these are composed of pallene protein, and they are not used in locomotion, but their main function is conjugation. Now we discuss about the ribosome. Ribosome, as we know, they are the protein factories. They are composed of proteins and RNA. Then mesosome. Mesosome is basically invagination or enfolding of the plasma membrane. As we know that bacteria lack all the membrane-bodied organelles. That's why mesosome is a vesicle, is a tubule, is a lamella that control different activities of the bacteria like DNA replication, cell division, respiration, and even though export of enzyme. So these activities are performed by the mesosomes. Then plasmid. Plasmid is basically double-stranded circular structure. This is extra genetic material, extra DNA. This plasmid have no concern with the growth and metabolism of bacteria. This plasmid is used in genetic research as a vector because this plasmid contains certain genes which have a high resistance power against the diseases, against the pathogens. Then comes plasma membrane that is composed of lipoproteins, and this is a semi-permeable structure that protect the bacteria from osmotic lysis and regulates all sort of transportation. Then cell wall. Cell wall is outside of the plasma membrane. This is made up of amino acids, sugar, cation. But cellulose is totally absent. Apart from these things, cell wall may contain peptidoglycan along with the teichoic acids, glycoproteins, and lipopolysaccharides. While we are discussing about the function of cell wall basically provides shape to bacteria and protection from the osmotic lysis and give rigidity to the bacterial cell. So these are the main function of the cell wall. While we are discussing about the flagellum, flagellum is basically, this is thin appendage, halo appendage that is originating from the cytoplasm, from the basal bodies. Whenever the basal bodies rotate, this flagellum moves, and because of this movement, bacteria locomote from one place to another place. So this flagellum helps in locomotion, and this is composed of flagellin protein. So while we are discussing about capsule, capsule is a protective sheath around the cell wall. This capsule may be saline capsule because this saline capsule provides a great pathogenicity to bacteria, and on the contrary, it also protects bacteria from phagocytosis. So this is all about the structure of the bacteria. So inshallah, dear students, we will discuss next chapter inshallah in next lecture. Thank you.